Welcome to Careers in Discovery, where you'll meet scientists who've forged outstanding careers in biotech and hear about what they've learned along the way. This podcast is brought to you by Singular, building brilliant biotechs. Steve Trim is the founder and chief scientific officer of Venomtech, a truly unique company that helps biotech researchers to work with venom-based assays. Steve talked to us on Careers in Discovery about his journey building a remarkable company, how he learned to be an entrepreneur and business leader, and his experience contributing to the Human Genome Project. This week on Careers in Discovery, I'm really looking forward to talking to Steve Trim of Venomtech. Steve, welcome to the show.
Yeah, thanks, Tom. Thanks for the introduction and the opportunity to have a chat.
Yeah, good to see you, Steve. And I'm really interested in your business and in talking about your career. Why don't we start by talking a little bit about Venomtech and the pretty unique proposition that you've got there?
Yeah, sure. So Venomtech are a very unique business. They've got a large USP, and that is that we provide venoms for principally drug discovery and biopesticides, but also a third market is cosmetics. And what we were doing, what we are doing, it's the same as what we were doing.
Yeah.
Is providing venom. So we've got a collection of venomous animals on site, which we manage to optimize their health and husbandry. We recoverably collect the venom, and I describe it much like milking a herd of cows. And the term is called milking when you're collecting the venom. And then we separate it out into its component parts, and it's understanding the biochemistry of those individual components and how they interact with receptors and ion channels and enzymes, membrane proteins, and all the things all the sort of cool biology within the venom, which then makes them useful as either tools or therapeutics or biopesticides, and cosmetic active ingredients being the third there. So, and it was set up to meet a need, basically.
Okay.
That in drug discovery, we call the low-hanging fruit, the easy drugs to develop have been done. So we're naturally, therefore, left with the tough stuff. And in pain and neuroscience, the small molecules are they struggle with selectivity because the ion channels are quite similar, whether those ion channels are controlling pain processes or cardiac processes or central nervous system processes. And obviously, you want your analgesics to just hit the pain ones and not the other ones. But that's really challenging. And venom peptides have a larger interaction surface, and a lot of them have evolved to target ion channels as a method of either capturing prey or actually causing pain and teaching predators not to eat you. And so by unpicking these individual components, we can actually find ones that are more selective and therefore potentially more usable as an advanced pain therapeutic.
Yeah, interesting. And I suppose that makes total sense as to why you would explore the uses of venom if the targeting is so good. So tell us about, I guess, the origins of this idea and where this business came from in the first place. So it's not the first place people would look for a therapeutic, I don't think.
No, that's very true. But there are therapeutics from venoms out on the market already, but I'll come to that in a bit. The idea from me, or the idea for Venomtech really came from me and a unique set of circumstances. So I was working at Pfizer as a biologist in pain and neuroscience, and looking for better analgesics and selective ligands for these. At that time, voltage-gated sodium channels that people are still looking at now.
Right.
So I understood the deep biology of what we were trying to do. I'd been working in drug discovery for 10 years, and understood that process, but was really frustrated that we weren't able to get hold of venom peptides in an assay-ready format. the academic literature was showing many of these peptides could be really useful for what we were trying to do, but actually sourcing them was very, very challenging.
Right.
I was the safety officer or safety delegate for pain therapeutics, and that involved toxins that we had been able to acquire and work with, things like tetrodotoxin from pufferfish, for instance. And the third component is that I understood the venomous animal biology because I've been keeping non-dangerous spiders and snakes as pets. So I still do, but that hasn't changed. And so when Pfizer decided that myself and my colleagues needed something new to do, when they downsized the operation in Sandwich,
Right.
I then sort of realized I had those key skills, and I knew it wasn't just a Pfizer problem. You know, the reason that we are still struggling to get good pain therapeutics out on the market is this problem, and it's across all pharmaceutical companies that we get incremental changes, but nobody's fixed all the problems. So that I knew that I wasn't just setting up a company to just supply one customer, because that's a foolish errand. I knew that it was a big market to try and do, and so I then went about, okay, well, how do you collect venom from a spider? Sort of work that out on the kitchen table, and I understood the need and, let's say, the safety, and then I then formed Venomtech in just under a month from leaving Pfizer, which was possibly a little too fast, but it is part of the story. It's what happened.
Yes.
And then I funded it through my redundancy initially, and then went to sort of grow the company from there later on, because my mindset is a scientist. Even 13 years later, I'm chief scientific officer at Venomtech, not the CEO, and that's deliberate because I'm still very much a scientist. That is my mindset. The business side of things I have to work on, whereas the science comes more naturally.
That makes sense, and I suppose it's a logical step to, if, as you say, if you're trying to find pain therapeutics, to think about, well, what are the things that cause pain, and what are the things that are very effective at that, and then, how do they do that, and I guess that probably reveals some mechanisms that you can perhaps
that's
leverage
part of it, but it also, another core part is that the evolution doesn't have a plan or a pattern. So when a venom causes an excitation of an ion channel or a blockage, that impairs a prey organism's ability to get away in either direction. And evolution is not fussy. If it slows the prey down enough to be caught, it works and it stays.
Right.
So there are ion channels that are involved in that have then evolved in humans and become part of pain processing, because we have, all life is sort of connected to one or at least a few forms, and so we share these common genes.
Yes.
And that's that sort of interesting serendipity that animals haven't really evolved to envenomate humans. That is, we're quite new on the evolutionary scene. The one exception that is spitting in cobras. There was a really good paper in Nature last year or year before, showing that actually the act of spitting does seem to be directly correlated with bipedal hominids.
Interesting.
But generally, venoms have evolved to be active on other species other than us. So it is a bit of serendipity we're making use of, and definitely venoms haven't evolved to kill cancer cells. There's no benefit in helping your host live longer. But it's something they do, and it's something that's useful in drug discovery.
Yeah, interesting. And you mentioned there's this gap in the market, and that was sort of initially triggered by a problem that you were trying to solve yourself, and a shortage that you identified. And I'm surmising here, so please do correct me if I'm wrong on this, but I'm assuming it's the combination of these animals need to be treated in a particularly careful way, But as well as then you need the scientific expertise to isolate the venom and isolate the active compounds and turn it into something useful that can be used in research.
Yeah, and that's exactly it. But
Right.
there are a few venom labs around the world, and most of them are focusing on treatment for envenomation. So they are
Mm-hmm.
looking at the challenge of the venom, and the only licensed treatment for envenomation at the moment is antivenom, so antibodies to those
Yes.
venoms. And in large these groups, that is their focus. And so they don't understand the nuances of drug discovery and the fact that we need to actually separate the venom out into its component parts, because most venoms have several hundred components,
Right.
and some it's a few thousand. And when you put all that mixture into a biological assay system, you're going to get the sum of all the parts and not see the bit that's interesting.
Yeah, I see. Okay, interesting.
So it's
So
that assay ready format that is the key bit
yeah,
of what we do with the venom.
yeah, interesting. We will come back to this. So you mentioned, Steve, as well, that of course, you were the founder of the company. You've taken a decision to remain the chief scientific officer rather than taking the CEO position. So tell
Yeah.
us a bit about that, and tell us a bit about what you're spending your time on, and what your day looks like.
Yeah. So for the first 10 years I was leading the company.
Mhm.
But it was clear that was a hard task, ultimately trying to do the science and the management and everything together. And so when we raised equity investment in 2020, on the back end of that year, part of that plan was to bring in a CEO.
Mhm.
And Paul Grant's the guy who brought in. He's been with us since then, leading the team, and he's an assay um, commercial guy, so come from DiscoverX amongst many other companies.
Yeah.
Um, and so I had that commercial expertise, and keen to take on the reins and the opportunity of the CEO. And it allows me to then focus on the science, because
Mhm.
a lot of our customers don't know they want venom, but they do know that they've got a problem trying to find a drug or a pesticide to do what they need to do.
Okay. Yeah.
And so I spend a lot of my time um, educating people in how venoms work and how they could be useful in their assay platforms,
Mm.
and then how they could be developed as drugs or biopesticides. And sometimes that's directly talking to the scientists, and other times it is writing scientific papers or books,
Mhm.
or presenting at conferences to get that information out there. And the other reason is that's what I would enjoy doing more.
Yeah.
The science. Both my wife and I sort of biology is a way of life. It's who I am. It's not
Right.
just a job I do. I don't switch off from being a biologist.
Yeah, of course.
And so being able to sort of focus on that science is the fun part of the job for me.
Yeah. And you mentioned earlier as well that you were already interested in keeping exotic animals, and so
Yes.
you've been able to combine that passion as well with this, which is, I guess, an added bonus.
Yeah, very much so. And a lot of people think, oh, I've got into it because of the pet keeping,
Mm.
and that's not quite that way around. The
Hmm.
passion is for the science and biochemistry. The pet keeping is an enabler that
Yes.
helped me solve that problem.
Yeah. Sometimes it's about the perspective at which you look from which you look at these problems, isn't it? And
Yeah.
it's all your experiences that add up to that. So yeah, that makes sense. Um, but us take it further back for a little bit, Steve, and talk a little bit about, I guess, the origins of your scientific leanings. And so, because I know you trained as a geneticist
Yes.
originally, but tell us a bit about, why science, why genetics, why drug discovery, why this career for you in the first place?
That's a great collection of questions, yeah. And for me, I can't define why science.
Okay.
It was just ever since I've had science, you know, come across in my path at school, it's been fascinating.
Mm.
I can define why genetics though,
Okay.
and that to me comes down to a single moment. Um, so sort of, just sort of at school, all science I was involved with sort of great at sort of lapping it up. But it was a program by David Suzuki, and this probably was going to be somewhere in the late 80s or early 90s, about the potential of the Human Genome Project. And I
Yes.
remember him standing there with a huge filing cabinet and pretending to have a printout of the human genome, which wasn't it wasn't reality then because the project hadn't got that far through, but it was about to be starting. And saying that these 4 letters encode everything that the human body does. And I was like, wow, and I need to know more about this. This is just absolutely amazing how a molecule makes everything that we perceive and see. And I need to know about this basically.
Right.
So then I went to do A-level biology, chemistry, and physics, and a degree in genetics at University of Wales in Aberystwyth. It's a fantastic location and a great genetics university there. And through that, we got exposed to molecular genetics, plant breeding, chromosomal research, and a whole range of things. And it was sort of clear that molecular genetics was really sort of my thing of understanding how those molecules are controlling the natural world. And what was really great opportunity, a little bit as engineered, I went out there and got it, but being offered the opportunity was the key thing.
Yes.
So I did my year in industry at the Sanger Centre on the Human Genome Project.
Wow.
Which sort of nicely tied up my inspiration for following genetics. And that was we were in a radiation hybrid mapping lab, so actually looking at where the bits of DNA are on the chromosomes.
I see. Yeah.
Rather than actually the full sequencing labs. And this lab was running year-out projects every year, basically. A group of undergrads would come in and add a bit more to the map. And so being able to do a little piece of that was fantastic. And then they just sort of reinforced that, molecular genetics is what I want to be doing.
Yeah.
I did my final year project on the control of a metabolic switch in Clostridium baehreinckii, which is a nightmare as an undergraduate to try and spell. Too many eyes in it, and an anaerobic bacteria. So there was a strong experimental discipline in having to work in the anaerobic cabinet. So because if you've forgotten something, you have to degas it for 2 hours before you can use it. So you get extremely disciplined in making sure you've got everything ready before you start.
Yes. Yeah.
And then, I applied for PhDs and industrial places doing applied genetics. Basically, it was clear to me that was what was driving me. I didn't mind whether it was going to be a PhD or in industry. I just wanted to do that science. And the job at Pfizer was the one that was most interesting out of what I got offered.
Yes.
And that took me down to East Kent, starting initially in the gastrointestinal team. We did a rotation through their pain therapeutics and tissue repair and genetic technologies, and all those 3 things to actually understand different parts of the business. And then, from that, I sort of in big pharma companies, you end up being sort of molded, obviously, where the need is. And pain and neuroscience was the sort of need that I was there for 8 years, and then that led me into toxins.
Yes. Yeah, absolutely.
And the world of a protein biochemist rather than a geneticist.
Yes, and it's interesting how often these sort of these turns occur in people's careers, especially in larger companies, right? Because there's so many opportunities to do different things. That you can follow both the need of the business and your interest to some extent.
Yeah, and it's both the things you can follow and things you have to because of strategic changes within the company.
So I suppose thinking about the time you spent at the Sanger and the time at which you were studying genetics, I guess if people are sort of fairly recently qualified scientists. The Human Genome Project is probably something they take for granted, right?
Yeah.
And there's of course still work to do on mapping the dark genome
Yes.
and all these kinds of
Yeah,
things.
yeah, yeah.
But I suppose the things that are building blocks of research today and the advances that have happened in genetics and now genomics and everything that's come beyond that will still be informed when you were learning about it. So tell us a bit about that.
Yeah, so that was really quite fascinating that it was coming
Mm,
by piece. And because the Sanger being one of the core founders of the consortium, they actually had a counter up in the reception that was counting the number of bases that had been sequenced.
I see.
Uh, um, and one of the things that happened in the year I was there, so that would have been 96, 97, I think.
Hmm.
Um, and we celebrated 100 megabases of raw sequence that had been deposited by the consortium,
Hmm.
which was a fantastic thing. And when you look at these, they call them flat files. It is literally just reams of text with just four letters in it.
Right.
No structure or annotation to it. But yeah, if you're experienced, you can see genes and you understand, start codons and stop codons. You can start to
Hmm.
see things in the almost like matrix style text, um, which is really quite fascinating.
Yes.
So I'm drifting off on other things a little bit, but then in much later, it was only about 5 or 6 years ago, I think. I was at the Wellcome Trust Centre in just down the road from King's Cross,
Mm.
and they actually have a printout in chromosome volumes of the Human Genome Project,
Okay.
and it's not quite as big as David Suzuki's big filing cabinet,
No, no, no.
but it is still it's still huge. And yeah, the fact the way that's sort of building and coming on, and then the when the Nature paper came out of the first draft of the human genome of going,
Mm.
yeah, I did a small part of that. There's minor bugbear that us year out graduates were not listed, but the author list was enormous.
Yes,
Um,
of course. And
and
it's
it
on
was
this podcast now, Steve. So it's
yeah,
in the record
I've
now.
got my claim. Yeah, I may have mentioned it once or twice before. Um, but yeah, actually, as the sequences come together, um, and you could see the ensemble database building. So the European Bioinformatics Centre is still closely associated with the Sanger at that site
Hmm.
in Cambridgeshire. And getting that annotation on the genes it's sort of really quite fascinating. And now we're coming into a new chapter where we're looking at the sort of it's either 10,000 or 100,000 genomes. I can't remember what that project is. Actually looking at individual mutations and how
Right.
what gives rise to the variety of the human condition, basically, and where there is mutations in the genes that are not very much normal, just causing differences that we see in protein expression
Hmm.
patterns, as well as those that cause diseases, and therefore gives us insights into drug discovery. And one of the great things that the Human Genome Project allowed us to do is that sort of the $1,000 genome, because we can now sequence a whole human genome for about $1,000,
Right.
but only because that Sanger sequencing template is there. But
Hmm.
these high throughput sequencing methods still can't tackle brand new genomes. They rely on templates. So that
Right.
because even though Sanger sequencing is seen as sort of old hat nowadays, but um, for new genomes you still have to rely on a little bit of that sort of sequencing. But it has allowed the sort of high throughput sequencing to understand diseases. And this is something that became very relevant in my career in pain at Pfizer.
Mm.
And some of the Pfizer scientists in the team we were working with um, were doing a genome-wide association study with patients with a congenital insensitivity to pain. Now, most people think that that is a good thing. If you don't feel pain,
Right.
surely that's great. But these people have a very short lifespan and have a lot of comorbidities, because when they rest accidentally on the cooker, shut your
Mm-hmm.
finger in a door, they don't feel the pain response, so they
Yes.
get maximal tissue damage, unfortunately. And so doing this sequencing understood that there was a mutation in the sodium channel nav 1.7. It's a voltage gated sodium channel that infers this congenital insensitivity to pain. But otherwise, these, apart from a lessened sense of smell, otherwise these patients were normal. Their cardiac function was fine, their cognition was fine. So it showed this was, this is the holy grail of pain targets, because if you have a mutation and it doesn't work, you don't have a side effect burden apart from your lack of pain.
Right.
And so then that led, when that paper came out, the whole sort of pharmaceutical industry in pain research to move to, okay, let's go and try and drug nav 1.7. And the challenge there is that small molecules of drug nav 1.7 also interact with nav 1.5, which is involved crucially in cardiac pacing, which is a quite dramatic side effect
Yes.
of a drug. And then you've got others that are involved in the central nervous system. And several venom peptides, particularly from tarantulas, because they are bigger molecules, they have a wider interaction surface.
Okay.
Some of them are a lot more selective to nav 1.7 over 1.5. And that then led into the toxins and venoms we were just sort of talking about, led to the idea that came into Venomtech.
Yeah.
And so it nicely links together that the human genome sequencing sort of gave that link into why I was then going to go and look at venoms and peptides.
Right, because without that, there's no way to understand why that
without
side effect happens,
that large
right?
scale sequencing, we wouldn't have understood what was going wrong in these people to actually then find ourselves with a gene target.
So for someone who was particularly interested in the applied nature of genetics, I mean, this must have been like a dream project to be. And I get the sense as well from, I've spoken to a couple of people who were either involved or peripherally involved in the Human Genome Project, and there's always a sense that everybody was incredibly enthusiastic about it. It was a project that people could see the application of it unfolding before your eyes, right? It must have been a great place to just, if you needed any more fuel for that, to get it.
Totally. Yeah, it was fantastic. And I say, because we had that counter, so every morning when you come into work, you could see how many bases had been put in the database overnight.
Yeah,
it was a 24/7 operation with the sequencing anyway.
Absolutely. And so you, as you said, you were looking at where to go next, and you were looking at furthering your academic career or going into industry, and you went to Pfizer. There aren't many bigger companies you could have gone to, especially at the time.
No.
And you were there for about 11 years, something like that?
Just under 10 years and about 4 or 5 months, I think.
So you've touched on some of the things through your time there that led to what you do now, particularly. But I suppose tell us a bit about the experience of being part of a company like that, and the things that you learned there that have been, useful for you in the rest of your career, and the way that experience has influenced how you do things today.
Yeah, definitely. It's a really great job. I wouldn't have chose to leave. And that's for many reasons, that the learning opportunities are huge out there. And where I got to sort of a senior and scientist level, that I was being presented with problems of biological problems of where is this gene expressed, or which genes are controlling these sort of pathways. But how I found that out was pretty much down to me in it, and that was really exciting, to actually go, okay, well, let's see. Okay, we've got a potential side effect of this particular drug. Well, let's see if that drug target is in those tissues where that side effect's coming in.
Yeah,
sort of looking that apart, and so the sort of freedom there to if you look at new techniques and go out and do gene chip analysis, and anything you need to do on that, then the opportunities for training were immense. And yeah, I just sort of definitely encourage people to just grab all those training opportunities, because you never know what's going to be coming next. And although my core focus was gene expression studies, later on I was doing drug discovery pharmacology assays,
Yes.
and doing some protein biochemistry, which was frustrating at the time because I wanted to do more DNA stuff. But
Right.
in hindsight, it was incredibly useful. And then things like sort of add-on tasks and stuff. We've all got other things that need to be done in the company, and safety is one of those key things. And I saw the opportunity as being the safety delegate as to sort of be able to sort of balance the books a bit, because people see the safety people as sort of coming in and just sort of restricting all the fun things you want to be doing. But on the other side of things, you want to not be injured from doing your work. There's a benefit.
Yes.
There's a benefit from having those processes and sort of being involved there to actually look at, okay, well, why do we need to work with this particular toxin, for instance?
Mm-hmm.
And how can we do this in a safe way? Then actually, let's restrict the amount we need, and all sorts of simple things like this. But also then making sure that everybody's up to date with the literature of what they're doing, what they're working with.
Right.
And yeah, it did. I didn't see it as the great opportunity it was at the time. It was another thing that took me out of the lab,
Mm-hmm.
but it was a core set of skills that when I was sort of forming Venomtech, to actually have worked with toxins. I'd worked with the Home Office,
Right.
and understood the biology of what we're trying to do, principles of separation, and the standard hierarchy of control of do you have to do it, and if you have to do it, can you have a physical barrier between you
Mm-hmm.
and the dangerous thing, and all those sorts of standard stuff. But it then gave me sort of a extra set of skills
Yes.
on the other side, and that then actually led us to a patent for a safe method of feeding venomous snakes.
Right.
And all I did was employ the hierarchy of control to venomous snake feeding. Is that when zookeepers feed tigers, they don't sort of walk in with a pile of meat on their back and the tiger in the cage. However, how people were feeding venomous snakes was a bit more sketchy, right, from my observational point,
Right.
because I'd been used to working with non-dangerous snakes, and
Yeah.
yeah, you open the cage and offer them a dead food item, and that's fine. Whereas with the venomous snake, it's a bit more dangerous.
Mm.
And so we just got a piece of coy plumbing, so a cylinder that bolted on the side of the tank. There's a slide gate on one end and the screw cap on the other.
Mm-hmm.
We can undo the screw cap, put the food in, screw the cap back up, and then open the slide gate, and the animal could get the food, and it was still contained.
Right.
And that was so revolutionary that the patent got granted. And we let it lapse because it was just a safety statement from our point of
Mm-hmm.
view that we're working differently with this. But had I not had that pharmaceutical health and safety role and training, I would have looked at other people and how they were keeping venomous snakes and just sort of just
Yes.
followed suit. But it
Yes.
actually gave us a completely new insight, a new way of doing these things.
Yeah, absolutely. And it's sometimes, as you touched on there, it's sometimes hard to join the dots forwards, right? Sometimes you can only join them in
Yeah,
hindsight.
yeah, yeah. And the other great thing doing drug discovery in big pharma is the research budgets are huge.
Sure. Yes.
um, that if there are things that will help you do your science faster and better, the answer is generally yes.
Mm.
Let's go and get it. And when you then set up a small business, that was not useful training
Right.
because it's a very different thing on the other side. You have to be a
Yeah,
lot more inventive, and things take a lot longer.
absolutely. And I think there's an emotional component to that, right? You really feel every pound that you spend.
Yes, particularly when it's yours in the beginning.
Yes. Yeah, absolutely. So yeah, moving on then to Venomtech. So you said earlier that of course you had your redundancy package. That was your initial investment into the business. I imagine that only went so far because businesses are expensive, especially research businesses. And you turned it all around very quickly, as you mentioned. You you also said you've taken on someone to look after the business side of things now, but that you were responsible for that for the first part of the business's life
Yeah.
as well. So I imagine there's lots of things that you learned during that time. But, your memories of the early days of the business. What are the things that stick out to you?
Yeah, that is thing. It's a steep learning curve, and it
Mm-hmm.
is quite exciting. And the things that just I thought had more to it. I think the fact that purchase orders and invoices, when it comes down to it, you just write it on Word. That's all that they're required.
Yeah.
And in the technology age that we're in, a letterhead is not as daunting. You had to go to the printers and get a stack made in the typewriter days. And people say, "Oh, can you send this on your letterhead?" And at first I was like, I don't have. Well, what's that? And you realize that it's just your logo in the header
Yeah.
of a Word document.
Absolutely.
And yeah, things that seem daunting were not. But the big thing, and it's a real cultural difference, is that if I was ever required to do anything that I hadn't done before at
Mm.
Pfizer, it would either come with an SOP or training, often both.
Mm-hmm.
Whereas when you're out in the outside world, it doesn't.
No.
And I think the first shock was the certificate of incorporation that comes through the post
Mm.
is just that. It doesn't come with a handbook.
No.
That you have to go looking to find out,
Mm.
well, what does it mean. Now I have when you're signing up for a company, it does tell you about the legal responsibilities that a
Yes.
company brings. It doesn't tell you how to do these things. It
No.
just tells you what. um, and you know, financial projections and stuff. You go to the bank when your redundancy runs out, and they ask for a business plan.
Mm.
Okay, well, I'll have to go and find out what one of those is there.
Yes. Yeah. Yeah.
And things like this. So um, my learning was quite steep because, and it's something I get asked to do lectures for universities and stuff from time to
Hmm.
time, quite often on business as well as the science. And my sort of top advice is, if you're setting up your own business, is get somebody else's money as well as yours,
Yes.
because it was just my money. There was no checks. I didn't have a business plan. I had a
Mm.
scientific idea. Turned out to be a good scientific idea, but
Hmm.
it was just a scientific idea. It was only when that money started to run out that I actually then needed to build a business properly
Yes.
to actually get other money in. And I think that was a big learning point for me, that if I'd um, known that from the beginning and then gone and got um, funding to go alongside my redundancy, it would have lasted longer, but
Hmm.
also we would have started from a much stronger point.
Yeah, and it creates some accountability as well, right?
Yeah, got somebody else to answer to.
Yeah, it's
And,
easier
and and
to.
somebody else has checked your idea that it is
Hmm.
actually valid. Whereas when you start it up with your own fund, that's not needed.
No, no. And I think if you find the right investors, of course, then they bring something as well as money, right? They will have a
Oh,
different
definitely.
perspective. They'll have experience.
Yeah,
They'll have a network probably. That there's the advantages of that as well. And I think
yeah.
yeah,
And yeah, one of the good things that kept me on the right track early on is that I knew a reasonable amount about what I didn't know,
right.
and I knew I had to go actively looking to find these answers. And you very quickly come to the point where you can either pay for the answer or learn it.
Mm.
And you can't learn everything ultimately. So, um, I was right from the beginning involved in going to local networking events, both scientifically but also more local business networking events, and talking to people about what I'm doing and actually finding out what other people do.
Right.
And I found a finance director who was really interested in what I was doing, the science of what I was doing. So much so to actually have a bit more philanthropic relationship to help me sort of build on actually the idea of getting a board of directors together,
Mm-hmm.
and stuff like that. So that was sort of started relatively early on in the first couple of years, and then it wasn't just me. I then had a team of people that were
Yes.
looking at the governance of the company, which again was all new new territory.
Yeah, of
Um,
course.
and by sort of networking and finding people and sharing what little I had to actually sort of try and build that, and then just make use of every connection I could find, and actively
Mm.
going out there and talking to people, and old school networking is really really valuable to find people that can help you out on working out what they want out of it, and if it works both ways, then you can build something together.
Yeah, and I think this is sometimes the bit that the sort of the MBA programs miss, right? Is that some businesses are very technical, some businesses are not, some businesses are very complicated, some businesses are very
Yeah,
simple, but ultimately they're about people in lots of different ways, right? So your customers are
yeah,
people, your staff are people, your board are people, your investors are people, even your investors.
yeah, and your customers are people.
Yeah, exactly, and I think that that's the bit that gets missed, I think, when you focus on these cold clinical analyses of a business plan or what have you. But having those things is important. And I think it's like you say, you can never learn everything as a CEO of a company.
No,
You've kind of got to learn enough to know what you need to know and what you need help
yeah, and where your
with.
gaps are.
And where your gaps are, and as well, there's always people who have a vested interest in making it seem more complicated than it is, so they can charge
Yeah,
you a lot of money for it.
yes, yes.
Right? So, I mean, any advice around sort of how you obviously networking's been a big part of how you've learned things. Anything else that's been particularly useful for you?
Um, yeah, so reading and learning, I'm a scientist. I'm used to researching
Mm-hmm.
stuff. The term gets banded around wrongly nowadays, um, but that old school checking the sources and reading um, valuable material, and cross checking it and stuff, to understand what you need to do, and listening to people again with that have that good advice, but also things like the enterprise hub. So the University of Kent
Mm-hmm.
enterprise hub was the first one we were affiliated with. So we started at the back of a pet shop,
Okay.
because that's where the animals were,
Sure. Yeah.
um, and a bit of kitchen workbench, to actually work with some venom on,
Mm-hmm.
and that was Venomtech version 1. But there's another opportunity that came up serendipitously, really, was the genetics lab at the University of Kent wanted someone to analyze whole genome data in birds and look at evolutionary genetics.
Mm-hmm.
And they asked me about it, and I was like, well, I have just started a business. I've got a lot on my plate at the moment, but then strongly suggested that I apply.
Okay. Fair enough.
um, and I was like, well, my money's only going really in one direction at the moment. This would help. There's a time constraint here, but I can help. So I then applied and I ended up getting a post. So it was a postdoc post, but I just called it a post because I'm not a doc.
Right. Fair enough.
Because my training is industrial.
Yeah.
Actually doing this avian genomic stuff, which is really quite interesting. And then was come home, have some tea, and then I was going to the pet
Mm-hmm.
shop to do Venomtech work of an evening. Um, and actually that then, because I'd been a member of staff at the university, it then unlocked the being part of the enterprise hub, which is there to help staff with their businesses. So
I see.
although a company Venomtech wasn't a spin out, because I was a member of staff, that then got us into helping people build the financial models and a bit more of the business part of stuff coming through that opportunity. And we get a lot of that now at Discovery Parks. We sort of moved full circle in 2014, I think,
Okay.
moving on to what is called Discovery Park, which is actually the old Pfizer site. And in 2020, we actually moved into the building that I left
Oh, really?
in 2010. And so the enterprise zones, and there's a few of them around that sort of are there to help small businesses, and the culture there is to help people learn about running businesses and have experts there of various different things. And so when we moved into Discovery Park, we realized some of our neighbors were experts in logistics. Now, shipping venoms around the world is hard work when
Yeah,
you've
of
had to work
course.
out what you're doing from scratch. Whereas when you find people who that is their job, that is their expertise, you can make that a lot easier. So, but then it's still networking. It's just not labeled as networking. It's still building those sort of relationships.
Yeah.
And going on that, also then the investment side of things. So very quickly realized that because venoms are complex mixtures, we need quite serious hardware to separate them out into the component parts and make them useful. But I couldn't see how the catch-22 was solved between needing the expensive equipment to fractionate the venoms and needing the sale of the fractionated venoms to pay for the expensive equipment. And it was through forming the board where it became clear that we actually needed to raise investment to actually bring in that instrumentation and ultimately adds value to the product as well as applicability.
Yes.
And when you've got such a novel and unique idea, there isn't a tried and tested course. People can't
No, of
look at
course.
what Venom Company Y has done and what they exited for and what they turned over because the data's not there. So you end up realizing that the investors are buying into the team and the trust of the team and sort of liking the idea of the science because we don't know, we've not made that future yet of what exit of a Venom Company achieves in the UK. And so that understanding that when you're talking to investors, it is a two-way street. You mentioned it earlier that they bring skills and network to you as well as money, and they also have to people you can get on with because you'll be selling part of your company to do that, and you have to be able to work with them, otherwise it's going to be a very difficult situation. And that was really quite interesting that share certificates also are just made in Word.
Yeah.
They're written down on a register at company's house. But there's nothing more to it than that.
No.
And that's really quite an enlightening part of the process.
Yeah.
And there's a really good book called Sapiens. I know it's behind me somewhere, and I can't think who it's by off the top of my head. But I think it is a scientist of some form, and it basically talks about it's basically human society about how everything we do is based on trust. That I say this is a pound, and you believe that is a pound, and you understand what you can do with that pound. But if you don't trust and don't believe it, then it's not actually worth it. And business, it completely is like that. There isn't Venomtech isn't a thing. It is a piece of paper that I've said this is Venomtech, and the people on the receiving end have gone, yes, we believe this.
Yeah, absolutely.
And when you start to understand that, it becomes less scary that these things are not are so sort of potentially fragile, I suppose.
Yeah, absolutely. And I think, you look at businesses that are well established, and especially if you're in the early days of a business, they seem so far away from you in terms of how they do things and the infrastructure they have. And the way they operate and all these kinds of things. But I think, it's easy to forget that they've got years on you in a lot of
Yeah,
cases, right? And they've
yeah,
made lots of little change. They've not one day been this tiny business and then immediately turned into this huge business. It's lots of little changes
yeah,
and lots of
yeah, yeah,
little improvements. And it's as you say, the things like share certificates, the things like and it's all becoming easier as well. The more technology gets
yeah,
involved. A lot of it's common sense, isn't it? But it seems a world away when there's so much of it.
yeah, yeah, very much so.
Yeah. So, perhaps for someone who's starting out on that journey, and this may be something you've mentioned already, or maybe someone who's just starting out in their scientific career, is there maybe a key piece of advice you'd share, career advice that's been particularly useful for you, Steve?
The key thing in people starting on their scientific career is experience
Mm.
outside of the academic world, um, because the academic qualifications and teaching and learning is absolutely vital,
Yes.
whether you're doing science or any other disciplines. But when it comes to the other side, there is a transition for a lot of practical reasons. And so for me, having a year out in my degree actually working in an industrial lab um, meant that that was the sort of top topic of conversation in interviews,
Right.
because they wanted to know about how I work in what they call the real world. And that was the sort of top thing for me. And I always have a lot of time for students that are looking for work experience,
Hmm.
because they need to understand how things work on the outside. So when you go and get your first job, it's not a total sort of
No,
eyes wide
absolutely.
open total shock.
Yeah.
You've had a safety net introduction into industry,
Hmm.
and that was really really valuable, and also a surprise benefit for me, which I didn't expect, um, was that the starting salary was higher because I had a year's
Right.
industrial
Okay.
experience, and I was like, oh, that's quite nice. I have
Yes.
that's not why not why I done it, but it was a nice bonus. That for me, I just wanted to, yeah, I'm just sort of that sort of sponge to sort of gain all the experiences I
Mm.
can. But I very much see, and COVID has been a great exemplifier of this. We've got a whole triage of students coming through that have had very restricted lab experience,
Yes.
and it's of no fault of their own or the universities. Um, and to try and then get some experience is really quite vital
Hmm.
for two reasons. One is for your own confidence and capability, but also you might start working in a lab and realize it's not for you.
True. Yes.
So if you've done that in a shorter term test environment, it's less tricky to then fix.
Yeah. No, absolutely. And it's not for everybody, right?
No, exactly.
Yeah, it's as with everything. I think you have to have a particular temperament to be an experimental researcher, right? You
Yeah,
have to be
very
able
much so.
to take the negatives with the positives, and that's true of
Yeah,
everything. But I
yeah,
think,
yeah, certainly.
particularly in that role.
And field biology appears really glamorous when you see it in the wildlife programs on telly. But there's a hard side to that job.
Yeah, absolutely,
And
absolutely.
so if you can get out there and experience it first, that you come into a
Mm.
career with your eyes open, certainly. So yeah, that's sort of a top advice for students coming in their scientific career is get lab or field experience, whatever your interest is try it out for real, either over the
Yes.
summer or a year in industry, and to actually test it out first.
Mm.
And if it's what you like, then you've got yourself an extra bit of experience, which is great for the CV. And if not, then you've got the opportunity to then, in the next part of your studies, to tweak what you're trying to do to fit things you like. Because my sort of driving force again for a career in science is realizing that you spend most of your waking life at work. So
Yes.
it's good to do something you enjoy doing, at least for most of the time.
No, absolutely. And I think, it's also, I guess, when you train as a scientist, you you of course learn some technical skills and you
Yes,
learn some domain specific knowledge, and that's a crucial part of it. I think what people miss sometimes is the transferable skills that they're picking up, the
yeah,
the way of looking at things analytically, the way of developing a method to come to a decision about
yes,
something,
yeah,
all those things that are the way of structuring an argument, all those
yeah.
things that can be applied to lots of different paths. So if you were to go and get that experience and decide it's not for you, you're not then stuck, right?
You've
You
not
can go
lost
and
anything. No.
adjust
Yeah.
your path and take those skills and find something else that you like. So I think that's important to.
Yeah, very much so. And then sort of moving on, you also asked about advice for people
Hmm.
starting their own business, and it is um again, you know, get experience, talk to people who
Yeah.
are doing it, and yeah, go to networking sessions and interact with people that are, you know, running small businesses. We've all got a story to tell,
Yeah, yes.
and usually happy to share that advice. Um, so you actually again coming into it with your eyes wide open, that there's a there isn't a there isn't a sort of nurtured test way into the companies that so much with um trades or sort of selling crafts and those sorts of things, and you're sort of a sole trader. There's a blurred line legally
Mm,
between you and your business, and it's sort of a softer transition that you can sort of drift in and out from. Whereas making a scientific company, it was very clear that my customers were not going to buy from a sole trader. It
no.
had to be a limited company.
Yeah.
And when the ink's dry on that form, there is a lot of legal responsibilities, and you
Yeah.
can't just stop. There is a process that you need to go down if you want to close that company, and that costs
Hmm.
money.
Yes.
Um, so it's difficult to just test running a company. So yeah, definitely go and ask people that do, and there's plenty of SME forums, and yeah, if you're
Yeah.
connected with the university, then they'll have sort of business departments to help on this side of things. But yeah, that was the sort of the shock that suddenly you had it, and you've got a legal responsibility for this thing.
Yes.
And yeah, it will cost you money to stop. You can't just go, actually, no, shouldn't have done that.
No, absolutely. And as you say, I think, you generally find that people are pretty happy to share, right?
Yes.
It's not quite as cutthroat and competitive an environment as it might seem on American TV
No, no.
programs. And my general experience with it has been is if you ask a bunch of entrepreneurs to tell you their entrepreneurial story, then you'd better have set aside quite a lot of time for that conversation.
Because A, they like selling the story, and B, there's
Yeah.
a lot to
Yeah,
a lot to tell.
exactly. Exactly. Um, so bringing us back to today, Steve, what's next for Venomtech? What are you guys working on over the next 6, 12 months?
Yeah, that's a really good question. And sort of, as a scientist, making future predictions was quite alien to me because I just saw it as extrapolating beyond the data. But in business, you have to make those plans. They may not be the plan that actually happens, but you
Sure.
have to be ready for things going forward. And for us, it is growth in terms of customer projects coming in, but also the market. So the biopesticide market is relatively new to us,
Hmm.
so about 4 or 5 years now, I suppose. But um, we still don't have the real density in that space. So it is generating more data, going out to more conferences in the biopesticide space to actually then by sharing the word of venoms and how they can be useful because they are biodegradable
Right.
and they don't shouldn't bioaccumulate and be more selective to then sort of build our capacity in that space
Mhm.
alongside what we're doing in drug discovery. And so we're very much in a growth phase. We're still only little. It may be 13 years along, but it's not a sudden boom industry. It is quite niche, and we're quite early in terms of this science. And one of the things that caught me out actually early on is that when I was leaving Pfizer, they were talking about being number one in biologics.
Right.
And I was like, well, it's perfect time to set up a company selling biologics.
Yeah.
But the devil was in the detail. Is back then they were talking about antibodies, not
Yes, I see.
peptides.
Yeah.
um, Whereas now in the last few years, COVID has really helped the biotech sector deal with new drugs,
Hmm.
like RNA vaccines.
Yes.
They were People were talking about them, but nobody really had any sort of traction in that space. And peptide therapeutics are again, people are talking about it, but the traction is only just starting to come through for
Right.
us. So it is we're very much in a growth phase. Um, Not so much with the species. We The library is fairly dynamic. We bring in new species where projects demand it.
Hmm.
But it's the application of those and moving through from an identified peptide to a drug-like molecule. So we've started doing pharmacokinetic studies in-house,
Right.
looking at plasma stability, thermal stability, synthetic stomach fluid and synthetic intestinal fluid to look at how the venom peptides survive in those quite challenging biological systems.
Hmm.
But that helps us understand how we can tune those peptides to being more drug-like. And so that's sort of the sort of growth area for us over the next sort of 6 to 9 months, which is seeing me out at a lot of conferences, doing
I'm sure.
a lot of shoe leather. But
Yes.
ultimately, I enjoy it, being out talking to scientists about science, but always having that commercial hat on that you need to try and sell some things as well. It's not just cool science.
Yes, that's true. That's true. Well, look, it sounds like an exciting time for you. We wish you the best of luck with it, Steve, of course. And thank you so much for joining us today.
No, thanks, Tom. Thanks for the opportunity to talk about what I do. It is a fun thing, and it's one of the sort of top difference from the farmer job is that the amplitude of the highs and lows are bigger in your own
Yes.
business than
Yes.
in farmer. Farmer is a lot more predictable. Um, and yeah, it's a really exciting place to be. So yeah, thanks for the opportunity to just talk it through, and yeah, I look forward to hearing the results.
Absolutely. Thanks very much for listening. Careers in Discovery is sponsored by Singular, helping you to build a brilliant biotech company. Biotech leaders spend far too much time, money, and energy on hiring and people issues. Head to www.singular-biotech.com to learn how you can recruit and engage your team more effectively so you can focus on developing medicines, treating patients, and saving lives.